Hello, everyone. My name is Arthur Luhur, and I'd like to take this opportunity to thank the organizers for giving DGRC the platform to present, update, and showcase our products today. Let's begin. DGRC has three main aims. The first two aims relate to the collection and distribution of research materials, and the last aim is to develop and test technologies for use and to help people in using them. This is the current DGRC setup. We have one director and three associate directors, assisted by a group of project scientists, research associates, undergrads, as well as a database specialist. On the next slide, I will show you the new plasmid reagents that we have acquired since last year when we spoke here. Thank you to all the donors. If you go on to our website, you'll be able to see all these reagents if you line up the numbers from the biggest number to the smallest number. In addition, we have added a further 1,500 clones to the human cDNA collection. Today, I want to use this opportunity to showcase our cell line collection, and I hope this is going to be useful for individuals, researchers who have never experienced or used Drosophila cell cultures. So Drosophila cell cultures were derived from immortalized cell cultures back in the 1960s, a lot of them. They were from embryonic sources, larval sources, and adult sources. And the cell lines listed on the left panel, they remain the most highly used insect cell lines today. On the right panel, you'll immediately get an appreciation that different cell lines have different morphologies, different growth patterns, and different gene expressions too. Before I forget, in the past year, we collaborated with the RRID database to assign cDNA clones all our reagents, cell lines, plasmids with RRID numbers. So make sure you include them when you write the materials and grants when using our products. With a collection of 200 plus cell lines, there needed to be an effort to characterize the cell line. One such big effort was the mod-encode project. And the end result of that particular project, if you look at Flybase now and ask a simple question, is the gene escargot, for example, which cell lines express it? We can easily find out that the ovarian cell lines have them, but most other cell lines do not. Now, I want to focus on one thing from the mod-encode project that I think is going to be important. That is the DNA copy numbers. I want you to appreciate here that this particular cell line 1182-4H, it was initially derived from a haploid animal, haploid source of material. So it was a haploid cell line even during 3 years of continuous culture. When we received it at DGRC, it was still a haploid cell line. Over time, by the time the mod-encode project ensued, this cell line had become minimally diploid. So the bottom line is that cell lines evolve. Now, contrast this cell line with a commonly used cell line S2R+. You'd immediately see that the number of chromosomes here are much, much more than the previous cell line. And in some cases, you have more than 20 copies of chromosomes, just like a human karyotype here. So this cell line, S2R+, is minimally tetraploid. Our catalog of cell line at DGRC, most of them are cell lines derived from animals of known genotypes. For the modified cell lines, we have three different classes. The first class is the cell line in which we have genome insertion sites so that you can put in DNA constructs of interest. So for example, here shown, you have CNS cell lines, embryonic cell lines bearing ADP sites for you to insert your genes of interest. And they are all at different locations, some of them on 3R, some of them on chromosome 2L. We are currently still enlarging this particular collection, and to add more cell lines. The next class of cell line is the endogenous gene tag lines. Shown over here in the live video is an S2R+ in which Polo has been tagged with GFP. One easy application is cell biology, where you can just observe, or you can couple this in a cell-based screen. The next class is a CRISPR knockout cell lines. Now, one quick application here, as illustrated by this publication highlighted by Houston et al., is to use this cell lines as a genetically sensitized background and coupling it with an RNA screen to look for any genes that you're interested in a particular biological process. And in their case, they're looking for drug targets for the TSC complex. One major development recently is CRISPR, and coupling CRISPR with Drosophila cell lines, we have the CRISPR-based screens. This workflow is from the DGRC at Harvard. I'm not going to go through in detail, but suffice to say, this is a very powerful method for functional genomics. There are at least 3 current papers now that have utilized this method here to look for genes of interest. And we can have a Q&A, talk more about this later. Another thing that is important is the TE transposable element profiling in cell lines. It gave us insights into the genome evolution in cultured cells. One finding here is that cell lines, they have increased TE activities compared to flies. Long story short, it became a method to authenticate Drosophila cell lines. So if you have a cell line somewhere in your freezer and you're not sure if it is truly an S2 cell line, reach out to us, we can help. Now, I want to switch gears and highlight some curious and interesting work using cell lines. In particular, in the study of cell migration, immunology, and metastasis. Shown in the next slide here is the video in which you have made a scratch on the growth surface, and within the course of 16 hours, cells start migrating to close up the space. Staying on the theme of migration, in 2008, the Simcox lab injected a cell line which overexpresses the oncogene RAS, an active version of it, and followed what happened in the animal a week later. So as you can see in the top right panel, you have an animal that is glowing green because the cells have proliferated inside the host. Fast forward a couple of years later, Hoffman lab looked at what happened to the cells when inside the animal. They found out that injected cells proliferated, and not only that, they induced new trachea development, presumably to support their own growth. They were able to isolate these cells and do transcriptomic analysis on both the host, the injected cells at day 0, and finally, the injected cells at day 11 to follow how they have evolved. Now, I will close by talking about a bit about future directions of DGRC. So I've highlighted some of the interesting ways people have used our resources. So we want to continue to modify existing cell lines for more utility and to create cell type specific cell lines, formulate new medias, and explore 3D cultures. Finally, I want to acknowledge the people who we have collaborated with, Doug, Shunhua, and Casey, Simcox Lab, Paramount, and Stephanie from Harvard, DGRC Lab, all individual donations, IU facilities,